Hello, I'd like to thank the organizers for putting together a great meeting. Today I'm going to tell you about the work we're doing to develop novel hematopoietic cellular therapeutics. Fate's been around for 7 years. We are a publicly traded company located just across the street, and we have about 57 employees. We're publicly traded, so here's the requisite forward-looking statement. And so in a nutshell, what we do is develop small molecules that program in new functionalities into cell therapies. And just to put an analogy that helps me explain this is think of it as an app for your smartphone where you load the app on and now your smartphone can do a new function. In a similar fashion, what we do is take hematopoietic cells, and in this case, shown here is on the left is a unprogrammed or normal hematopoietic stem cell, and we expose it to small molecules for 2 hours right before it's administered to a patient, and we program in a new function into that cell. And in this case, what's shown in green is the expression levels of an important homing protein called CXCR4. And in a transplantation setting, this is important because that extra protein level helps get the cell back into the bone marrow niche, and it accelerates immune reconstitution. And it's a big deal because it gets people out of this period of time where they're really susceptible to infections. And similar to the app analogy, you have a lot of different apps for your phone to do a lot of things. We could put in different small molecule cocktails. So in this case, this is a combination of 3 small molecules, which again, for a human HSC, that following the treatment, we've dramatically upregulated a protein called PD-L1. So this is a very important immunoregulatory protein. And we're taking that red cell now into preclinical models exploring use in inflammatory and autoimmune disease. So just the idea that we're using small molecule treatments before the cells are administered to the patients to add new functions to those cells. So again, we are focused on the cells of the immune system, and we're spent a lot of time studying CD34 cells, NK cells, and T cells. And there's 3 main areas that we're applying this sort of cellular programming. The first is in the area of transplantation or chemotherapy-induced severe infections. And basically what I'll tell you about is we're treating donor grafts with small molecules to accelerate immune reconstitution with the goal of reducing infections. The second thing I'll tell you about is we're working with NK cells and T cells in the immuno-oncology space, and here we're really focused on increasing the persistence and the tumor-killing properties of these immune cells. And finally, that red cell I just showed you, these again, we're exploring the use of a chemically programmed CD34 cell for autoimmune disease. So the first one is the immune protection area. So chemotherapy-induced severe infections is a really big problem. And this is especially the case for patients undergoing hematopoietic stem cell transplantations where you receive full myeloablative conditioning, which basically wipes out your immune system, leaving you very susceptible to infections. In fact, 80% of patients will experience a severe infection the first year post-HSCT, and 30% of these patients don't make it through this first year due to infections. So our goal is to create a way to improve reconstitution by using this ex vivo small molecule treatment. What's shown here is a unit of cryopreserved cord blood. And standard of care, there's been 30,000 of these procedures performed over the last 20 years. Standard of care is to take a frozen unit of cord blood, thaw it, wash away the DMSO, and immediately administer it to a patient. After it's been infused IV, it takes 26 days on average for the new immune system to come back online. And again, it's in this window of period that people are susceptible to infections. So we've tried to do is stick as close to standard of care as possible while still changing, profoundly changing patient outcomes. And so what we do is we take the cryopreserved cord unit, thaw it, wash it, and then we incubate it in a specially designed media with, in this case, it's a molecule called DMPG2. It's a stable derivative of prostaglandin E2. Following a 2-hour incubation, we wash a cell away to remove the median modulators and the enhanced cord blood units administered to the patient. And again, the expectations are we'll accelerate immune reconstitution and reduce infections. So what's shown here is in a cord blood unit, about 1% of the cells are these hematopoietic stem cells. There's a variety of other very important cell types that are administered into the patient that actually provide immune protection involving specifically NK and T cells. So what's shown here is a gene expression plot after the 2-hour treatment showing that hundreds of genes get dramatically upregulated. So CXCR4 is upregulated 20-fold during this 2-hour incubation. Other genes involved in proliferation and survival are also upregulated. And that is the cell that's administered into the patient. But if you take a look at the other cell types in the cord blood unit, so we're treating the entire cord blood unit during this procedure, and we're also having profound impacts on specifically the T cells and the NK cells. And we're studying this very carefully to see can we make those cells perform better during this first critical window post-transplant. So getting into a clinical setting, so again, we take a cord unit, we treat it to impact the 34s, NKs, and T cells. We wash the modulators away, administer the cells to an immunocompromised patient with the goal of accelerating immune reconstitution, reducing infections, and improving survival. So we're about 2/3 of the way through a phase 2 study testing this idea. It involves 60 patients that are randomized 2-to-1. We're in 11 of the top transplant centers. And again, these are folks with hematologic malignancies receiving a cord blood transplantation. We recently revealed the data for the first 30 patients. We'd like to wait until all the patients are out 100 days past the procedure just to get a snapshot of how they're doing. And one of the things that we saw, which was encouraging, is a 6-day acceleration of neutrophil engraftment. So again, as I told you, under myeloablative conditioning, it takes 26 days. So we've brought that in, and so that was a good sign that the modulation is doing what we wanted to do to the hematopoietic cells. Another thing that we observed is a interesting reduction in severe infections involving bacterial, fungal, and viral. I think part of this is probably due to the earlier neutrophils, but the viral infections that would point more towards an NK or T cell mediated effect. So we're studying this very carefully. And as we head towards the end of the year, we're going to reveal data on, hopefully we'll be completing enrollment, and we'll do another data cut, and hopefully these trends will continue. So earlier in our development pipeline, we're also creating a similar approach to help specifically designed for mobilized peripheral blood. So mobilized peripheral blood is a predominant source used for transplantation. Over 70% of allo procedures involve this as a cell source. In this case, the key unmet need, infections are still a problem, but the key unmet need here is high levels of acute GVHD. So we went and designed a combination of small molecule modulators where we now treat the mobilized graft, again, right before it gets administered to the patient. But here the goal was to, again, make the immune reconstitution happen quicker, but also to see if we could mitigate GVHD. And also the trick here is you have to preserve the health of the T cells because they're instrumental in fighting the cancer in this post-HSCT setting too. So you can't just kill the T cells as your solution. Here's just a quick snapshot of data where we're now taking this exact solution that we're putting our IND together, which we're going to file at the end of this year. But you could, again, take in a mouse model, mismatch model of GVHD, you could pre-treat the cells, administer them into irradiated mice, and then you could track GVHD scores as well as survival. We could also introduce tumors into these mice and just confirm that in addition to seeing lower GVHD, you still have the ability to fight the tumors, which is really the right balance that we want to achieve. So really quickly, we're also using the small molecule programming approach for immuno-oncology. And again, the goal here is in cancer immunotherapy is we want to make these cells work better at taking the tumors out. We're doing a collaboration with Jeff Miller at University of Minnesota. He's identified this type of NK cell called an adaptive NK cell that has high levels of CD16 and it has epigenetic signatures that are very analogous to cytotoxic T cells, and they have memory-like features and they persist much longer than normal NK cells, and that's the real challenge in initial clinical trials with NK cells. So we're working with him to again go through small molecule screening to push cells into this adaptive phenotype, and we're taking these into in vivo models to show that we can increase the persistence and the efficacy of the tumor-killing properties of those cells. This summer, we also announced a collaboration with Therapeutics, where, again, we're applying our small molecule screening engine on their CAR T therapies. What we want to do here is they're interested in increasing the persistence of their therapy, and in many cases, we screen for small molecules that skew the T cell population to more of a memory stem phenotype that, in our in vivo models at least, will have increased persistence. In the next generation of this idea is we'd love to figure out how to target CAR T cells effectively to solid tumors. So both of these projects involve patient-derived T cells. We spent the last 7 years working hard on generating therapeutic-grade IPSCs. And what we think this is going to, and what the direction we're using this is to create off-the-shelf immunotherapeutics. And the key advantage here is that being at the IPSC levels, you have the opportunity to do multi-step engineering. And in contrast to autologous cell therapies where you're doing engineers on a patient-by-patient basis, this allows you to do your engineering once, sequence the genome, confirm that it's safe, and then differentiate it at industrial scale to large quantities of NK and T cells. And the dream here is to really, I think the autologous cell therapies have shown the promise of this field, but I think that for this really to go to the next level, we need to figure out how to get scalable off-the-shelf manufacturing, and we're working really hard towards that. So in summary, again, we're using this idea. We're focused on developing novel hematopoietic cell therapies. We're using small molecules to enhance the function. In many cases, these treatments are done right before the cells get administered to the patient. We're excited about the work in the transplantation area, where we are using small molecules to accelerate the recovery of the immune systems to prevent infections, and in the case of mobilized peripheral blood, reduce the incidence of GVHD. Immuno-oncology, we have some great partners where we're taking, today therapies and making them better by doing our small molecule programming to improve the persistence of NK cells and trafficking of the CAR Ts. And I think we're very, again, as I said, we're very excited about second-gen version of these cell types using our iPSC platform, where we could combine the power of genetic engineering with the small molecule programming to really take it up a notch. And finally, we're exploring the use of small molecules to create these CD34 cells that express high levels of immunoregulatory proteins such as PD-L1, and we're testing these in a variety of inflammatory and autoimmune models. So that's a quick snapshot of what we're up to, and I'll be happy to take any questions at the break. Thanks.